Familiarity with the tools and methods of any branch of science is essential for proper understanding of the subject. We will review some of the more common methods used to study cells, tissues, and organs. Light microscopy, electron microscopy, histochemistry, and cytochemistry, immunohistochemistry, and cell culture are described briefly below. Light microscopy is the most common technique in histology research. The routine histological preparation for light microscope examination is a paraffin section stained with hemotoxylin and eosin, He. Procedures of preparation are as follows: obtaining the specimen, fixation, dehydration, clearing, imbibing, sectioning, He staining. Obtaining the specimen. In general, tissue should be as fresh as possible. Experimental animal tissues are obtained immediately after the animals in question have been killed. The tissues taken are cut into small pieces. Fixation. The tissue blocks are rapidly fixed with chemical fixative in order to preserve their structural organization. Two of the most commonly used fixatives are formalin solution and Boyne's solution. Dehydration. Fixed tissues are immersed in areas of ethyl alcohol solutions, thereby increasing concentration until absolute alcohol is reached. As a consequence, water in the tissue is replaced by alcohol. Clearing. Xylene is commonly used as a clearing agent. Dehydrated tissues are passed through successive changes of xylene until all the alcohol is replaced by xylene. Since xylene has a high refractive index, the tissue becomes transparent. Imbibing. The tissues are then passed through two changes of melted paraffin to ensure the replacement of all xylene with paraffin. Subsequently, the tissue and the paraffin are poured into a mold and allowed to harden. The tissues are now embedded in the paraffin. Sectioning. Usually, sections 3 to 8 micrometers thick are cut with a microtome and mounted on glass slides. When the sections are dry, they are ready for staining. He staining. Sections are then stained with hemotoxylin for 1 to 10 minutes, with eosin for 2 to 3 minutes. This is a paraffin section obtained from the kidney, stained by He. Shown in the red box is a renal tubule. We can see the epithelial cells in cuboidal shape. The cytoplasm is stained pink. The nucleus is stained blue. Here you must know some professional terms. Basophilic. Hemotoxylin as a basic stain binds to the acidic components of cells and tissues, which then show a blue color. Such components, such as nuclear chromatin, are said to be basophilic. Acidophilic. Eosin as an acidic stain binds to basic constituents and gives a pink color. Such constituents are termed acidophilic. Other special staining, silver staining, stains neurons, neurofibrils, epithelial, etc. Such structures are termed argyrophilic. Aldehyde-focusing staining mainly stains elastic fibers, elastic membranes. Toluidine blue staining. Stain cells such as mast cells. Toluidine blue is blue in color, but the cells are stained in purple. It is called metachromia. Addition to above routine paraffin sections, frozen sections of fixed or unfixed tissues may also be cut with a freezing microtome or in a cryostat. Since preparing frozen sections does not subject tissues to dehydration, clearing, and embedding, some chemical compounds and enzymes can be better preserved. Electron microscopy includes transmission electron microscope, scanning electron microscope. Transmission electron microscope. The electron microscope permits visualization of the internal structure of cells. Structures identified with the electron microscope are called ultra-fine or subcellular structures. This is transmission electron microscope. This is the ultrastructure of the hepatocyte. We can observe the nucleus, mitochondria, and other organelles. Scanning electron microscope. The electron microscope reveals three-dimensional surface, architectural cells and tissues. This is scanning electron microscope. This is three-dimensional surface structures of blood cells. E is erythrocyte, G is granulocyte, M is monocyte, L is lymphocyte, P is platelet. Histochemistry and cytochemistry combine histology and cytological methods with chemical and biochemical methods and reveal the chemical composition of tissues and cells in situ. This permits a precise interpretation of chemistry of cells and tissues in relation to their structure. Many substances, for example, proteins, amino acids, nucleic acid, lipids, carbohydrates, ions, catecholamines, and enzymes, can be detected in situ by histochemical and cytochemical techniques. PAS reaction is used to localize polysaccharides. The arrow indicates the glycogen in hepatocytes. Sudan staining shows lipid droplets in adipocytes. Enzyme histochemical staining shows the enzyme in the cell. Immunohistochemistry uses labeled antibodies as specific reagents for localizing tissue and cell constituents, antigens in situ. Immunohistochemical staining shows that the smooth muscle cells are acting positive. Cell culture is a technique for the study of living cells. Isolated cells are cultured in a sterilized cultured medium at the appropriate temperature. Since the medium contains essential nutrients such as amino acids, vitamins, etc., cells and tissues grow in vitro. This technique is quite useful for detecting the effect of various reagents on living cells.